Neuroscience has a pretty broad scope. The brain, the retina, spinal cord, peripheral nerves, and neuromuscular disease. And so our next talk is going to be around neuromuscular disease with Mark Rich, who is an MD Ph.D. at WashU and then neurology training at Hopkins. He's one of the interesting folks. You know, when you collaborate in neuroscience, you can do it as a clinician collaborating with a basic scientist. In Mark's case, he really does a little bit of both. So he does some clinical work and mostly lab work. And very interested in critical illness, neuromuscular disease, particularly myopathy. So, Mark? Okay.
To move this down as much as I thought I would have to, Brian. It's kind of, maybe we're not so different at Honey. Anyway, so we're going to go a bit full circle. Dr. McDonald talked about ion channel diseases that are genetic, and that is the vast majority of ion channel research. What I'm going to be talking about today is a very different idea of what I'm arguing is a new type of ion channel disease that has previously not been recognized. And I'm going to also argue it may be the most common ion channel disease that's out there. And it's a disease where you have dysregulation of genetically normal ion channels causing a problem. And I think Dr. Schweitzer's talked too, where you have the genetics coming into the normal brain and the interaction. I'm going to argue, why do you have seizures sometimes and not others? Well, your normal channels are constantly being modulated. And that's the sort of mindset I want to get you into here. Whereas normally, the research into ion channels is you have mutations. Ion channels have one behavior. And when they're mutated, they behave abnormally. I'm going to argue something very different. Normal ion channels can behave abnormally, depending on their situation. I want to start with a case that I'm going to present, that I think this case this person had this syndrome that I'm going to be talking about today. So I saw this patient about 10 years ago. He's a 22-year-old male who got severe pneumonia and overwhelming sepsis, so infection throughout the body, almost died. He spent 2 weeks in the intensive care unit, was barely pulled through. Once he recovered enough, they took off the neuromuscular blocking agents. Those agents are given so that you can ventilate the lungs in very sick people. The trouble was, after they pulled off the neuromuscular blocking agents, he remained in a coma, completely unresponsive to deep pain stimulus. And so they called us in neurology to really evaluate, should we pull the plug? Did something horrible happen, and should we just let him go? Because there was basically he was brain dead. So we in neurology, when we come to see patients in the intensive care unit, we were called in both to look at why was he in a coma and why was he paralyzed, because you couldn't get any responses from him. So I'm going to start off with looking at why he was paralyzed, and we're going to talk about the coma later. Okay? So when patients have weakness in the intensive care unit, there are two main causes we think of that are acquired causes of weakness. And one is called critical illness polyneuropathy, which is a nerve problem that happens when you get overwhelming infections. This was first described about 30 years ago. The reason for this is not well understood at all. For some reason, the nerves die. Basically, our understanding is evil humors. We're still at that stage. The trouble is it leaves patients paralyzed for weeks to months afterwards. And so you pull the patient through, you kind of have this win from the intensive care unit standpoint, and the trouble is they have a horrible quality of life afterwards. Okay, and so this is really one of those unanticipated consequences of medical advances. These diseases I'm talking about didn't really exist 30 years ago when the intensive care units weren't good enough to pull people through. But now they're a major cause of disability. And the number one complaint of patients who survive intensive care units is weakness. Okay? The second cause of weakness is called critical illness myopathy, and it's a muscle disease that also affects patients. So what we do when we're called in as an EMGer, which is what I am, we do electrical studies of the nerves and muscles, is we do studies to tell these two diseases apart. And let me just go over what we found in our patient. So we basically shock the nerves, And then we measure the responses either from muscle or the nerve when we measure the electrical responses. And in this patient, we found no sensory responses in his body. Okay, so all sensory nerves, the idea would be they're dead. Okay? We also found no motor responses in his legs and tiny motor responses in the arms. So both of these top two are exactly what you would expect if he had the neuropathy. So both the sensory and the motor nerves would be dead, and so you won't get sensory or motor responses. But in addition, we did a test that I basically started 15 years ago in these patients, where you directly stimulate the muscle to test whether it can respond electrically. And before explaining why we would do this, I'm just going to give you the results. Okay, so we're just going to move forward. So shown here are the patterns that you get with direct muscle stimulation in patients who have neuropathy, the nerve problem, versus myopathy, the muscle problem. Okay? So let me just, oops, boy, this is sensitive. So what we do on the bottom here, this is the response when we stimulate the nerve in this patient, and we get this tiny little electrical response. Just so you know, normal is this big. Okay, so this is about 1/10 of normal. And that's because the nerve's dead in this patient. So you stimulate the nerve, you don't get a muscle response. We're recording the electrical response from muscle. But if we bypass the nerve in this patient, and now we directly stimulate the muscle, as we turn up the current, we can get a nice big response. And that's because the muscle's happy and healthy. Okay, it's just disconnected from the nerve. So this is the pattern you would see with a neuropathy. In a myopathy patient, the problem is in the muscle. So when we stimulate the nerve, we again get a small response. But when we go to the muscle, we can't bypass the problem. We turn up the current here, and we still get a small response. Okay? And this was proof that in these patients, the muscle actually could not respond electrically. If you biopsy the muscle, it's alive. But when you study it electrically, you can't get anything. And so this is basically the way I discovered that muscle in these patients has lost the ability to make action potentials. Okay, and I want to stress this is in genetically normal people. These are not people who have some inherited channelopathy. So I'd argue this is the first description ever of a severe ion channelopathy in genetically normal people. So this is what we found in this patient. He had the myopathy pattern. Okay, so now let's go through things. So if we just focus on the direct muscle stimulation, he has critical illness myopathy. He has electrically inexcitable muscle. Well, now we go, though, he didn't have sensory responses. You can't get that from a muscle disease, so he must also have critical illness polyneuropathy. Okay, so he has to have two separate problems. In addition, something I didn't mention at all is when he got EKGs, or electrocardiograms, he had very low amplitudes of those, and his heart wasn't contracting well. This often happens in sepsis. It's called high output cardiac failure, sepsis. So we'd have to say his heart was also not working well for a separate reason. And finally, he's in a coma, and we don't know why he's in a coma. So his brain is also failing for a separate reason. Okay? And this is what is known in septic patients as multi-system organ failure. All the organs are failing, we don't know why. Okay? And that's where we're at. I'm going to leave that right now as he has multiple problems in all the organs, they're all unrelated. I want to now do a little bit of a digression that will actually change the way we think about this patient. I'm going to talk about work we've done in rats to understand why the muscle becomes electrically inexcitable. Okay? So this is going to be changing gears a little bit. So what we're now showing you is we can cause critical illness myopathy in rats, and I'm going to skip over how we do that. And the advantage of that is we can dissect out the muscle and then use sharp electrodes to impale the fibers and record detailed electrophysiological studies that we can't do in patients. And let me just go through what we can do. So here we are, we've poked into the muscle fiber, and we have a resting potential of minus 88 millivolts. On the y-axis is voltage. On the x-axis is time. And shown here is a bunch of superimposed records of different current injections we've done. So for example, we inject some current right here, we turn it off, we don't get an action potential. We do it again, and I'm superimposing that, we turn up the current even more, we go up to here, no action potential. Then we turn up the current ever so slightly, and boom, we get an all or none action potential all the way up to here. This is normal muscles behavior, all or none action potentials. Well, what happens in a disease? In the disease on the right, these are 3 different muscle fibers, and we're doing our same superimposition of our current injection. This fiber has all or none action potentials, but look, they're much shorter, okay, they're much smaller than the normal, so even this guy isn't completely normal. And over here, these 2 fibers have very abnormal patterns. Here we have these very short and fat action potentials. Now, I don't want to say short and fat is bad, but in the world of action potentials, this is definitely not what you want to be. And here, as we turn up the current, we get not an all or none action potential. It actually gets bigger. So this is very abnormal behavior. And finally, this fiber is just dead to the world. We inject more and more current, and nothing happens. So this was proof that we truly have inexcitable muscle, and so something has gone horribly wrong. To cut a long story short, we have found out that the problem is actually with a specific protein, the sodium channels. And so I want to do a brief review of sodium channel behavior before going on. Sodium channels can exist in 3 states. Normally, when you're sitting at your resting potential in a fiber, they're in the closed state, so no sodium flows. When the voltage depolarizes, that causes the channels to open, and you get your action potential. The positive sodium rushes in, depolarizes more and more channels open. The interesting thing about sodium channels is after opening, they don't just close again, they move to what's called the inactivated state. And in the inactivated state, they're both closed and you can't open them again. Okay, so they're temporarily useless, basically. After all the channels are inactivated, your action potential ends, and then because of that, you go back to minus 85 millivolts, and that negative voltage causes you to move from the inactivated to the closed state. Okay? Well, so what would happen if all your channels were in the inactivated state instead of the closed state? You would become electrically inexcitable. Okay? And that is what we have found the problem is in critical illness myopathy. And so let me just show you on the next slide, summarizing a great deal of work. This is the sort of curve we can study. So on this y-axis is your percent of channels that can open to make an action potential. So up here when we're at 100%, all of the channels are in the closed state, nobody's in the inactivated state, and you get 100% of your current. Down here, all of your channels are in the inactivated state, and so nobody can open. So what we're plotting is, as we change our voltage, what percent of channels are in the closed versus the inactivated state. So here, everybody's still in the closed state, but now as we depolarize more, we start to inactivate, and we get less and less current, till at about minus 70 millivolts, we only get half, or 50% of the maximal current. Okay, so this is the voltage dependence. We can say the voltage dependence of inactivation of these channels in normal muscle is about minus 70. Well, what we found in the disease is our ion channels, our sodium channels, were inactivating at minus 86 millivolts. So we had shifted the voltage dependence of the channels. And again, I want to stress this is genetically normal channels. And we've done a lot of experiments showing that it's the same protein behaving differently. Okay? Well, so now we have this information that we have an ion channel problem affecting sodium channels in our muscle. Let's go back to our patient and see how that makes us think about him. Okay, so what we now would say is he has a sodium channelopathy making his muscle inexcitable. Well, you have sodium channels in your sensory nerves that also give you the action potentials. They're actually quite similar to the ones in muscle. Well, so what if he has a sodium channelopathy in his nerves too? Now we could say we have one problem doing both the muscle and the nerve. Well, what about the heart? The heart actually has the same sodium channels as some of the ones in muscle. So could he also have a sodium channelopathy in the heart? Oops, what just happened there? Not even, I didn't back and away.
Connection.
Is it the connection? And if the patient was in outer space, how do we, AV help? I could have figured that out. I just didn't want to. Right, I'm feeling rather stupid at this moment, but we'll carry on. Okay, so you could have the same sodium channelopathy in the heart. And finally, what about the coma? What if you have the sodium channels in the brain are not that different? So we could start to hypothesize we could have one problem, in fact, in one protein, and suddenly this patient doesn't have all these complex different things. He has one problem. What if we could fix that? Well, so let's go through tissue by tissue and see how much proof do we have for that. Okay, for the past 5 or 6 years, I've been spouting this, but finally this year, I have proof, I think, to back up almost all of these statements. So let's go through. Let's first do sensory nerves. And this was done in collaboration with Tim Cope's lab, where they can impale single axons of sensory nerves in living rats. And so what we did is we made rats septic. We basically go in and poke a hole in the gut and sew them back up. It's like a gunshot wound to the abdomen. And what the Cope lab did was record action potentials, and shown here are action potentials in a control, 3 different axons. This horizontal line represents 0 millivolts. The action potentials go up above 0 in most of the axons, and a few just barely miss. And in the septic rats, the action potentials were smaller, particularly focused on these sorts of action potentials. And here's just a histogram of the action potential amplitudes. There's control. Most of them are 50 millivolts or bigger. And in the septics, there was about a 20 millivolt shift. They were much smaller. Okay? And there are a number of reasons you could have unhappy action potentials, but one would be if your resting potential was different. So we took axons that had the same resting potentials, did the comparison again, and we basically got the same answer. And again, to cut a long story short, this data and other experiments we did strongly suggest that it's a sodium channel problem in the peripheral nerve. Okay, so now we have strong evidence for a sodium channel problem in the peripheral nerve and the muscle. What about the heart? Well, so a number of years ago, we collected EKGs from patients serially who had sepsis. Since this is a 12-lead EKG, and I'm no cardiologist, so all I focused on is look how big the QRS complex is, all these lines. For those of you who aren't medically oriented, just how many squiggles are you seeing in general? In the septic case here, 7 days of sepsis, you can just see there's not much here. Okay? 2 days later, not much. Patient recovers. The EKG recovers. Okay? Now, there are a lot of different reasons you can get this, okay? And this was all brought up when we submitted this paper, and it's a long battle, lots of technical issues. So this does not prove you have a problem with electrical excitability of the heart. So recently we moved to rats, made them septic, took out pieces of the beating heart, and recorded action potentials. And here's what we found. These 2 solid lines are the action potentials from control heart, okay? And the 2 dotted ones are from septics. And you can notice the action potentials are smaller. And the action potential in the heart's more complicated than the other tissues. The rising phase is sodium current, and this plateau phase is calcium current. Well, the deficit, if you actually look at this slope right here, it's in the rising phase, which is the sodium channel phase. And we plotted that here. You can look at the rate of rise as an estimate of sodium current in the controls. Here you are. And in the septics, it was down. We looked at the duration as a measure of calcium current. It was not different. So it appears that we have a selective reduction of sodium current in the heart. And in the rat, we could actually selectively block sodium channels in control hearts and show that it made the heart beat less well. So just like sepsis. So I would now argue that we have evidence strongly suggesting that the reason the heart doesn't beat well in sepsis is the same sodium channel problem happening in skeletal muscle, happening in peripheral nerve. Finally, we've moved to the central nervous system. This is again done in the Cope lab. The recordings are all done by Paul Nardelli, who's sitting in the front row over there. And what we did is we made rats septic, and then Paul went in, poked individual motor neurons, and we recorded how they respond to injected current. So in a control motor neuron, if you inject current, the motor neuron fires repetitively. As you turn the current up, it fires faster and faster. And notice it never pauses. It's very regular in its firing. In sepsis, you have this much slower rate of firing. It's irregular. It pauses here. As you turn up the current, you just can't get the same sort of firing response. And on the right here, we have a 4.5-second pulse. Controls always fire the whole way through. This motor neuron basically poops out for 1.5 seconds, just sits there. This is very abnormal. Again, suggesting that we have a problem with excitability in the central nervous system. Now, in the motor neurons, we haven't proven yet that it's sodium current, so we're not quite as far along as in other tissues, but that's an ongoing area of research. Well, so I want to close now, coming back to our patient and talking about things. And so we now have strong evidence, rather than just hypothesizing, that during critical illness, you have a sodium channelopathy affecting skeletal muscle, peripheral nerve, heart, and the central nervous system, motor neurons. I think this channelopathy can account for the weakness, the cardiac failure, and the coma in patients like this. So multiple different problems. To me, the most exciting possibility then is if we could fix this, figure out what's wrong with the sodium channels and fix it, we might actually be able to correct most of the multi-system organ failure that happens in sepsis. Now, I can't explain kidneys, lung, and liver, so there's still, gotta admit, there's some tissues that I'm little bit. But at least heart, peripheral nerve, muscle, and brain is a good step. So let's go back to the patient. What did happen with him? So consistent with what I would have predicted would happen with the sodium channel problem, he recovered fully, went home normal, from basically being brain dead. So that's how I want to end here and see if there are any questions. Yes.
Hi, Cassandra, neurologist, and State Talk. Mechanism.
So we worked tons on the mechanism. I can tell you many things it is not. And we are finally close to some mechanisms. We have some tantalizing leads, but in the past I've thought I've had things as well. I can tell you it's not the type of sodium channel expressed. It's not the sugars attached to the sodium channels. It doesn't look like it's phosphorylation of the sodium channels. So sodium channels, unfortunately, are this big, nightmarishly complex protein. We now finally have some hints that nitric oxide signaling may be involved, and in the maybe next time, several years from now, I would actually have that. But to be honest, despite years of work, we've ruled a lot of things out, but I don't have the answer yet. But we'll see. Yes, Jeff?
Sepsis is a very complicated model. Do you have any feeling for what aspect of sepsis it is that induces this?
Yeah, so the question is, what aspect of sepsis induces this? As you said, sepsis is so complex. I've decided to take the approach, there are lots of labs working on the whole cascades of all the inflammatory factors in sepsis. My view of that is there are a lot of people working on it. It's so complicated. I'm choosing to work backwards. So once we can figure out what's wrong with the sodium channels, the exact signaling pathway, like let's imagine it does turn out to be nitric oxide synthase, making nitric oxide. I would then try to study that and work back in. But given the complexity of sepsis and all the trials that have tried to intervene in sepsis by blocking certain inflammatory pathways, generally have all led to worse mortality. I'm trying to stay away from that and work backwards from the things we know. Yes?
One comment. I think that one of the reasons why you may not have an explanation for other organ failure is it may be secondary. If you have heart issues, there's a possibility of perfusion of the other organs, maybe a problem causing failure of kidneys or
yeah, that certainly could be.
So that's one thing to look at. The second thing I was going to ask is, in disuse atrophy, what kind of electrophysiological findings do you see?
So if you just denervate muscle, which causes it to be inactive, and you do this in rat, the muscle actually stays electrically excitable and the sodium channel behavior does not switch. The model we use for this in patients, they get high-dose corticosteroids and neuromuscular blocking agents. So we do corticosteroids with denervation. And somehow the two of those together cause this bad reaction where the sodium channels shift. But it seems like inactivity is a key risk factor. I want to say that is one of the things that causes this problem. But alone, it's not quite enough. You have to throw something else bad in the mix. Either sepsis, corticosteroids, or something else with the muscle inactivity, and then you get the problem. But inactivity is part of the problem. Yes?
So, Josh Hoppers, Neurology, Neuromuscular Medicine. So just focusing on the critical illness myopathy aspect of this, how would a sodium channel defect account for the pathologic finding?
Well, so, yeah. One thing that someone in the field has noticed, I've glossed over a huge number of problems in the muscle disease. There's actually selective loss of myosin, there's massive atrophy of the fibers, and these chronic problems bother the patients a lot. So I usually try to mention that, but since I was trying to go quickly. Here's a theory I have, that since I was just saying muscle inactivity seems to be a bad risk factor for this. If you become normally when you denervate muscle, you develop spontaneous activity, right? You tick, tick. It's almost like muscle just can't stand to be inactive completely. Well, in these muscles, when you have the sodium channel problem, you are inactive completely. And I think that might almost be a bad thing. Muscle can't stand that. It even starts to die. Muscle's undergoing apoptosis and the loss of myosin. I think that it's possible that the sodium channel defect might be the first thing and then triggers a bad cascade. It may not be true, but that's one of my arguments why I'd like to treat the sodium channel thing first, see if the other stuff gets better. But it could be separate, to be honest. All right, one more question, then people are probably getting tired. It's getting late. Yes?
A couple of questions from a clinical perspective. You present a case of someone who, by your assessment, was clinically brain dead. A situation where we all, as neurologists, see this on a fairly regular basis. Certainly that clinical brain dead person is also not moving. So
Right.
the issue related to that makes it difficult. Any biological marker, I mean, you were studying the patient for purposes of trying to understand aspects of why he wasn't reacting, why he wasn't moving. Anything that you could, having worked in a long-term, low-level rehabilitation facility for a few years and observing patients who had been assumed to almost have been at a clinically brain-dead level, and within 6 months to 8 months, again, returning to a high level of functioning, we made the assumption that those issues were related to a regaining of neuroplasticity and the ability to resuscitate. It raises certainly this question that maybe there is a different mechanism that is causing the change that we're assuming is tissue damage, but maybe modification of tissue that is correctable in a different fashion enables these people to recover. So 2 parts. How would one make the assessment from a biological way at the time you see these patients? And 2, long-term, therefore, predictability for recovery.
Well, so what I can say is in this case, to me, when we studied him, the fact that his muscle was electrically inexcitable made me think he had a good prognosis. It's kind of counterintuitive. If we'd have gotten a good response from muscle, I'd have been like, this isn't good. But the point being, if your muscle's electrically inexcitable, we know that's going to recover. Right? Because the muscle, basically, when you withdraw the problems, the muscle does very well. So the reason I think we were getting almost brain death in him is that we know we have electrically inexcitable peripheral tissues. That's enough to explain his clinical syndrome. That suggests, well, the central nervous system could be doing well. Then one more side. I'm now getting into the realm of anecdote, but 2 times walking in to see these patients who've been almost brain dead, you walk in the room, they're awake, and from a neuromuscular perspective, it's like, oh, the sural nerve response is going to be back. So the peripheral nerve responses recover in parallel with their waking up from the coma. Again, this isn't science anymore, but to me it's very tantalizing that when we see the peripheral nerve responses come back, they wake up. And so, again, I'm an EMGer, so I'm very biased towards nerve conductions. I would argue that if you had these evidence of peripheral problems with excitability, that would make me more optimistic for recovery on patients, because then we have an explanation that's not structural. And once you have an explanation that's not structural, it can recover, is my view. All right, one last one, then we probably should stop.
John Terry from Neurointerventional Critical Care. You mentioned that inactivity is a risk factor for this problem. And typically, when you see this clinically, it takes some time to develop. A person doesn't get septic and become paralyzed right away. Have you ever looked at a paradigm where you stimulate the muscle?
So there's actually a group in Germany that's very big on this, stimulating the muscles, and they're showing that you can prevent this. To me, the trouble from the clinical perspective, the muscles we care about most are breathing, right? Because the reason they're stuck in the intensive care units or on the ventilator. Stimulating the diaphragm, to me, that's a dicey. So you might be able to do some limb muscles and keep them moving, but they'll still be stuck in the intensive care unit. So there's evidence that if you stimulate the muscles, you can prevent it. And in fact, I just have been in contact with this group saying, do you really think this will be any use clinically, though? Because the diaphragm's the muscle we care, and the intercostals. And so I'm not so sure how helpful that'll be clinically. All right, we should probably stop. Thank you.